afternoon. Thank you for joining us and welcome to the 19th Annual Borlaug Memorial Lecture. Today, we come together to honor Dr. Borlaug's remarkable contributions to the fight against world hunger and to celebrate those who continue his mission. This event is made possible through a collaboration of the World Food Prize and the College of Food, Agriculture, and Natural Resource Sciences of the University of Minnesota. The organizing efforts for today were led by 3 student representatives from the departments with which Dr. Borlaug had deep ties. Forest Resources, where he completed his undergraduate degree, Plant Pathology, where he earned his MS and PhD, and Agronomy and Plant Genetics, where he served as faculty. These efforts were supported by advisors Mary Buschetti, CPAN's Alumni Relations Director, and Bob Stupak, Agronomy and Plant Genetics faculty member. Please allow us to introduce ourselves as your hosts and student organizers. My name is Lauren Doherty. I'm a PhD student in the Applied Plant Sciences program.
Hi, I'm Kat, and I am a PhD student in plant pathology.
Hi, my name is Clara. I'm a master's student in the Natural Resources Science and Management program. We would now like to welcome Gary Muehlbauer, Department Head in Agronomy and Plant Genetics, to the stage to share opening remarks for today's special memorial lecture. Give a warm welcome to Gary.
Thanks, Kat. Good afternoon. Welcome to the Borlaug Memorial Lecture and the Emerging Leader in the Plant Sciences Award Ceremony. I want to thank the organizers of the event, Lauren Doherty, Clara Von Dolan, and Kat Topham, and the advisors, Mary Buschetti, who seems to be involved in everything on this campus, and Bob Stupak for their outstanding work to host this event. This is the 19th year of the Borlaug Memorial Lecture and the 12th year of the Emerging Leader in Plant Sciences Award. It seems just like yesterday that we started this event, and it's been quite amazing that we've kept it going for many years, and that's really a tribute to the student effort over all these years. So it seems like time has really flown by as we've gone past these last 19 years. The annual Borlaug Memorial Lecture commemorates the contributions of the University of Minnesota alumnus, Dr. Norman Borlaug. Borlaug made to fighting hunger and celebrates those who follow in Borlaug's footsteps. Norman Borlaug was an agronomist and plant breeder trained in the Department of Plant Pathology here at the University of Minnesota. He's widely known as the father of the Green Revolution, who developed high-yielding, short-statured, disease-resistant wheat varieties. His work significantly increased global food production, saving millions of lives by helping countries in Mexico, India, and Pakistan avoid mass starvation. For his efforts in combating world hunger and promoting world peace, he was awarded the Nobel Peace Prize in 1970. This lectureship provides a unique opportunity to meet and learn about his work from a current international leader who will be introduced in a few minutes. In addition to Dr. Borlaug, plant scientists at the University of Minnesota have a long history of contributing to many important discoveries, including pioneering the use of genetics and breeding, regenerating corn from tissue culture, identifying physiological races of wheat stem rust, and developing the honeycrisp pattern. Even though university faculty have made these types of important discoveries that have resulted in significant financial and environmental drivers for farmers in Minnesota and around the world, the primary legacy of professors are the people they teach and mentor. The Emerging Leader in the Plant Sciences Award Ceremony is one of the most inspiring events of the year as we celebrate the excellence of an alum of the University of Minnesota and indirectly recognizing the legacy of the faculty. Over the years, the committee has done a terrific job identifying the emerging leader in plant sciences, because when I look back at the previous 11 award winners, they have all continued a steep trajectory and are making major contributions to the plant sciences and to agriculture. Today, we will be recognizing 2 brilliant scientists, Amy Jacobson, a 2014 PhD graduate of the Applied Plant Sciences graduate program here at the University of Minnesota, and the Borlaug Memorial Lecture speaker, Dr. Arshnee Moodley, a microbiologist who works on antimicrobial resistance at the International Livestock Research Institute. With that, I welcome you again to the Borlaug Memorial Lecture and the Emerging Leader in the Sciences Award Ceremony, and I'll turn the podium over to Kat.
So I'm not Kat, but thank you, Gary, for the wonderful introduction. As we gather to honor Dr. Borlaug's profound contributions, we are reminded of the lasting mark he left on Minnesota and the world. In his honor, October 16th is officially recognized in Minnesota as Dr. Norman E. Borlaug World Food Prize Day. We are grateful to have Commissioner Tom Peterson here today to read this proclamation. Please join me in welcoming him to the stage.
All right, this is an interesting microphone, but we'll make it work. Again, I'm Tom Peterson. I have the honor to serve as Commissioner of Agriculture for the state of Minnesota, and want to thank everybody in the committee that puts this on. I've been able to be here a few times and kind of been on campus all day today on doing some other meetings, and always impressed with everything that goes on here. And it's always, I always think, as Commissioner of Minnesota, I travel around a lot, represent the state, and we always, brag about the university, and we brag about Dr. Borlaug. It's kind of interesting right now. I don't know if people saw this, but the U.S. Mint just released last week their commemorative innovation coins for this next year. They'll be available January 1st. And we, both Iowa and Minnesota, tried to claim Dr. Borlaug, and we lost. And it's kind of a bummer. I hate to say that in this room, not to be a downer, but so Iowa coin will depict Dr. Borlaug, and that's good because we want to continue to do that. Minnesota, actually, our innovation coin, and we worked on this, and it was kind of funny, but it's refrigerated trucks, and so you can get a coin with a refrigerated truck that says Minnesota on it. But it also just shows innovation and how important that was to the whole system. But it's been kind of interesting this last week because I got a text from the Iowa commissioner, and he said, well, I said, well, we'll both claim him, and it's very interesting. But I just had to share that. So my job today is to I also want to welcome Dr. Moodley and look forward to listening to her talk too as well. I get to read proclamations, one of my greatest jobs as commissioner, and I did remember my glasses today, so I'm going to go through that. So, whereas Dr. Borlaug was born March 25th, 1914, on a farm in Iowa, he received his Bachelor of Science degree in forestry, though, from the University of Minnesota in 1937, and then started with the U.S. Forestry at stations located in Massachusetts and Idaho. He then returned to Minnesota and received his master's degree in 1941 and his doctorate in 1942, both in plant pathology from the University of Minnesota. And I always think this is interesting, whereas Dr. Borlaug was a varsity wrestler at the University of Minnesota and helped introduce the sport to high schools throughout Minnesota through exhibition matches, as well as refereeing the Minnesota State High School Wrestling Tournament, which is a big deal. For his lifelong advocacy for the sport, he was inducted into the National University of Minnesota, Iowa, and Iowa High School Wrestling Hall of Fame. So quite a career. Whereas in 1944, Dr. Borlaug accepted the appointment as geneticist and plant pathologist to organize and direct the cooperative wheat research and production program in Mexico, where he was immersed in scientific research and genetics for the next 16 years. He was extremely successful in finding a high-yielding, short-strawed, disease-resistant wheat. And whereas Dr. Borlaug's agriculture research in Mexico improved crop management practices and transformed agricultural production during the 1940s through the 1960s and implemented throughout North America, Asia, and Latin America. And whereas 1970, in the middle of his career, Dr. Borlaug was awarded the Nobel Peace Prize for his work in agriculture, which resulted in saving hundreds of millions of human lives by preventing famine, alleviating hunger and malnutrition. And his work and that of his colleagues was responsible for saving hundreds of millions or more in the second half of his career. And whereas when unable to persuade the Nobel Committee to develop a prize just for agriculture, Dr. Borlaug founded and developed the World Food Prize Foundation to annually award a prestigious prize for extraordinary efforts in food security. And whereas Dr. Borlaug was recognized with his nation's highest science and civilian honors, the National Medal of Science and Congressional Gold Medal, the National Academy of Sciences Public Welfare Medal, and he is the only person in history to have earned these awards, along with the Nobel Peace Prize and the Presidential Medal of Freedom. And whereas Dr. Borlaug dedicated his life to serving humanity until his death at the age of 95, his legacy will live on as an inspiration to young people, as a reminder of the importance of agriculture production and a model of significance the food plays in ensuring world peace. Now, therefore, I, Tim Walz, Governor of the state of Minnesota, do declare, it says Sunday, October 16th, but I'm not sure that's right, but it's we'll go with it, 2025 as Norman Borlaug World Food Prize Day in the state of Minnesota. So thanks for being here and look forward to our lecture. Thank you.
Thank you for reading the proclamation, highlighting Dr. Borlaug's journey and inspiring. As part of this event, we want to celebrate up-and-coming leaders in the plant sciences who helped to uphold Dr. Norman Borlaug's legacy of innovation and dedication to solving global challenges through agriculture. Today, we have the privilege of presenting the University of Minnesota's Award for Emerging Leaders in Plant Sciences, which recognizes an outstanding alum for their impactful and groundbreaking research in plant sciences, such as plant breeding, genetics, agronomy, or plant pathology, while also demonstrating leadership, social impact, and international influence. This year's recipient is Dr. Amy Jacobson, and to introduce her, I'd like to welcome Dr. Tavore Abadi, a retired distinguished laureate at Corteva Agriscience. Please join me in welcoming Tava.
Thank you, Lauren. First of all, I want to thank Lauren and the organizer for inviting me to come from Iowa to actually introduce Amy. I actually wrote the nomination, so I feel proud that she's getting the award. And Amy, I will do a more formal introduction and then a more personal component of the introduction. Amy holds an undergraduate degree from Cornell in 2011. It's not close enough.
Yeah, I think even closer.
Okay, even closer. Okay. And during her undergraduate, she did 2 internships in breeding in Pioneer, in corn breeding, and an internship in Wageningen with Fred Van Nieuwek. And her project was the Boyce Thompson Institute with Tom Burnell. And her PhD, she did it here, and she got the PhD in 2014 in working with Rex Bernardo. And that same year, she was hired by Pioneer at that time, and as a molecular breeding scientist, and then as a discovery scientist. It was a dual role. And she progressed into that career through a role of breeder analyst, and then more recently, she has taken the role of corn breeder in Marion, Iowa. And she has had significant contributions, both during her academic career as a student through papers and presentations, but more significantly, for me at least, she is a co-writer on the patents of 3 very, very important hybrids in North America. And she has been mentor in the CFANS program and on the Borlaug Scholar program, and she is also a member of the Borlaug Scholar Committee. Due to all her contributions in Corteva, she has risen to the level of laureate, and which is a very high recognition within the organization for the contributions that she has done both internally and externally during her career. And in Corteva, she is a leader and an inspiration for younger scientists, and especially for younger female scientists their possibility to actually join careers, especially in the area of field breeding. which is an area in which Amy has a lot of passion. And on a more personal level, I would also like to say the things that I wrote on my nomination, right? And I interacted with Amy during her role as chair of the plant, the plant science symposium that was held here in Minnesota. That is a series that started in Minnesota and grew little by little to actually cover or reach around 70 organizations around the world. And the contributions of Amy and her colleagues at that time was to embrace the program into putting the program at an international level. So I mean, they did two important contributions. They actually worked on actually facilitating the connection across organizers across 3 continents, to put together a report about the program. And they also published a paper on their work on the program. A paper that they didn't know at the time, but for me, it was very consequential because it actually saved the program itself, right? I was able to use that paper published by students as a testament of the importance of the program and the impact that the program was having on students. And the last component is this component about leadership that I think is important to actually highlight in the case of Amy. Amy, I was the chair of the laureate committee in our group, and it was an honor to actually see Amy recognized as a leader, as an example, as in breeding, and in the way in her career, how her career went from an analyst and a person making emphasis on statistics into appreciating and in a way loving the field work and being integrated and becoming a reference for other breeders in the organization. So with all that, I will leave this to Amy. And Amy, congratulations once more.
Thank you, Tabaret, for that introduction.
Amy is going to present on Zoom, so just give me a second to get everything sorted.
And Lauren, let me know when you can see my slides and if you can hear me okay. Okay, great. Thank you, Tabaret, for that wonderful introduction. I feel incredibly honored to receive this award this year. And today I'm going to be talking about going from data to yield and utilizing predictive breeding in corn. So Tabaret did a lot of this introduction already, but I got my bachelor's at Cornell University in 2011 and my PhD at University of Minnesota in Rex Bernardo's lab in 2014. Right after that, I started with DuPont Pioneer as a discovery breeding scientist, and I was doing a lot of genomic selection work and how we can improve our modeling. And then once Pioneer became Corteva, switched over to being a breeder analyst, doing more analytical work. But I really wanted to go back to the field and to be able to utilize this analytical work in my breeding program. So earlier this year, I became a corn breeder, and I'm focusing on the 108, 110 maturity. And I'm located in Marion, Iowa. So I'm in Eastern Iowa. And I've been there my whole career. And
Sorry, I'm going to interrupt you for a second because you're on showing the presenter mode. Hope it doesn't echo too much.
Are you able to see my slide?
It's still in presenter mode.
Let me just.
There we go.
Yes.
That's good. Yes.
Oh, okay. Okay. Still able to?
All right. Yep, you're good.
Okay. Oops, sorry. Okay. So I have a picture here of the research station where I'm located, and we have our research plots kind of located here. And then we also have yield test locations all over the state and over the Midwest. So looking into a hybrid breeding pipeline. So at Corteva, we are working on improving the best inbreds that we can. And once we have the best inbreds in both heterotic pools, we combine those into creating the best hybrids that we can. So we do inbred development. And then once the inbreds are developed, we combine those and start looking at different hybrid combinations until finally selecting to become a commercial product. So that's looking at a funnel, we have many candidates at the beginning of the funnel, and we start narrowing that down to fewer and fewer candidates through genotypic work and also through phenotypic work, which I'll talk about. So when we look at this funnel, we start with creating new genetic recombination on the inbred side. So creating new genetic recombination through crossing inbreds to each other. And then continuing to test them and narrow down that funnel to then testing hybrids and narrowing those hybrids down to eventually commercial release. And we continue to recycle germplasm as we do this. But at the beginning of the pipeline, it's such a wide pipeline where we aren't able to phenotype everything out in the field. So instead, we utilized genotypic work. So we have molecular markers, which are very cheap. And we can predict the performance of these new individuals without having to test them out in the field. Testing in the field and phenotyping in the field is very difficult, it's very time consuming, and it's very costly. While markers are very cheap and we can do them very quickly and get those results very quickly. So at the beginning of the pipeline, we're relying a lot on this genotypic information. And then as we narrow down that pipeline, we can start utilizing more phenotypic information and rely less on those predictions and that genotypic information since we have more data from the field. And as we're making decisions throughout the field, throughout the years, these are the type of decisions that we're making of what crosses should we make as we start to create new genetic recombinations. What crosses should we make to make that genetic recombination? What progeny should we select as we narrow down that pipeline? What hybrid should we create after we create the best inbreds and then we cross them to create the best hybrids? And where do our hybrids work best? And we can utilize predictions at every step of this pipeline. The first being through breeding population predictions of what crosses should we make. As we narrow down what inbreds we're selecting, we can use progeny predictions. And then we can also use predictions on hybrid creation of what hybrids should we create, and also where should these hybrids be placed based on environmental characteristics. So we utilize predictive breeding in every step of our pipeline. So what is prediction-based breeding? So prediction-based breeding utilizes an observed trait out in the field. In this example here, say we're looking at yield. And we have the genotypic information or the marker information. We can create genetic signal by combining those two and create marker trait associations. And that creates our estimation or training set. Then we have untested lines, which only have that marker information and do not have that observed yield or that phenotype from the field. But instead, we can predict the performance out in the field just based on that genotypic information. And then the breeders are given that information of how would that predictive ability look? So in the different traits that they're selecting on. In this example here, say the breeders are selecting on yield, on moisture, plant height, disease traits, root lodging. They're able to utilize this predicted performance, weigh those traits, and then make those selections just based on the predicted performance from the genotypic information. So we did a study within Corteva where we wanted to look at the power of predictions as we're utilizing this predictive breeding. How much can we trust that? So we had a study a few years ago where we had many populations and many individuals within that population that we had an experiment, and we utilized that experiment for 3 years. So the same individuals were grown for 3 years at the same locations for those 3 years. And we wanted to see how that observed yield would compare to the predicted yield. So in this example here, we have our observed yield from year 1, created that estimation set by combining that observed yield with the genotypic information, and then predicted back on itself. So then we got the predicted yield. compared that to the year 2 observed yield. Again, the same individuals, the only thing that's different is the year here. And we wanted to compare that observed yield to that predicted yield. So on the y-axis, we had the observed yield from year 2. And on the x-axis, we had the predicted yield. And we have a correlation of 0.86. So that's a pretty good correlation for a trait like yield, which has a lower heritability compared to other traits. But 0.86 is still a pretty good correlation, just comparing our observed yield to what our predicted yield and how we're able to utilize those predictions. Again, utilizing that same experiment, but this time just looking at raw data. So we have year 1 on the y-axis, year 2 on the x-axis, and just raw data. So just the raw data of the yield from the field. And here we have a correlation of 0.73. And then we can start improving that correlation from year 1 to year 2 by looking at by adding in phenotypic adjustments in the BLUPs. So the correlation of the BLUPs from year 1 to year 2 is 0.86. But we can also improve that even more by utilizing GEBVs, or the predicted performance based on our estimation set. And here we have a correlation of 0.96. So you can see the correlations really start to improve as we improve the experimental design and analysis and by adding in marker trait associations. So this gives us better decisions and reduced noise, since we're able to buffer out that year effect and that environmental effect and also that management effect across the environments that we're testing in. And so utilizing these GEBVs really help the breeders make better decisions because they're able to buffer out a lot of that G by E and that G by Y, that year effect and that environmental effect, as compared to just using the raw data or even just using the BLUPs. So once we have that prediction, moving one step forward is looking at algorithmic enhanced decision making. So breeders are making a lot of, or at least corn breeders are making a lot of decisions this time of year. There's a lot of data that's coming in, and they're looking at a lot of data at once, trying to make decisions of what should move forward in their breeding pipeline. So they're able to look at field data, look at blues, look at BLUPs, look at the prediction data. There's a lot of data that they have to ingest in a small amount of time. So if there's ways that we can help improve that to make better decisions and to help save time, that would definitely help with the breeding pipeline. So we started using machine learning strategies. So we're training models based on what breeders' decisions were made based on a large amount of data, and if we can model what those decisions are and model what traits that they're weighting or down weighting. Can also start creating risk versus reward thresholds, where we're combining, looking at different traits that have risk versus reward and have thresholds for that. And also an optimized selection index based on what breeders have selected and what they would select in the future. So we go from looking at a lot of data to looking at much fewer data columns and make better decisions on that just by utilizing some AI in breeding. But it all starts with good phenotypic data. So we can't have good models without good phenotypic data. In this example here on this top left picture, we have a field that received a lot of rainfall right after planting. So you can see there's a lot of variability within this field looking up above from this drone image. In this example here, we had some disease come in, and you can see there's some plots here that are starting to die a little early, and that can cause some variability within the data too. This picture in the lower left is the Boreas wind machine. This simulates a windstorm, and then we can go through and collect root lodging or brittle snap data just from the simulated data that we're able to get here from this wind machine. So instead of waiting for these windstorms to happen, we're able to simulate that and collect data from that. And then it's also much cleaner data since we know it's the same wind speeds that's going throughout the field. While in a natural storm, the wind speeds can be very variable throughout the field. And then this last picture here is a LiDAR image, and we can start looking at new data utilizing that. So for automation and next generation data collection, I showed some images on that last slide taken by drones. So we're able to take a lot of different traits from the drone. This picture here is looking at soil monitors, so we're able to add some of that information into our modeling. Some VR goggles here is we're able to utilize that for decision making and for seed preparation. Some time-lapse photos as we're going through the growing season, and also utilizing other automated ways of taking data. So in this example here, we have Spot the robot going through the field and giving us a different view than what a human would if they were to go through the field, or a different view of if a drone was taking an image from above the field. And can get more images going through the canopy rather than looking from above the canopy. So I've been able to utilize all this work in my breeding program now. Like I said, I switched to becoming more of a field breeder earlier this year. So I am targeting mainly the Iowa, Illinois, and Indiana environments and creating hybrids for farmers in those regions. So some of the key traits I look at is yield, dry down, disease resistance, root strength, brittle snap, and also stress resistance. And there's a lot of opportunity for breeding in this area. There's a lot of corn that's grown in this area and a lot of high yielding corn that's grown in that area. It's a prime location and right in the middle of the corn belt. Some of the challenges are stress testing with different diseases that occur throughout the years, and also stress testing as we have different environmental conditions throughout the year. And like I said, I'm located in Marion, Iowa, so I'm right in the middle of my breeding target zone, and I go to many different locations throughout that area to see how the hybrids are adapted to those environments. So just building on Norman Borlaug's legacy of improving yield, disease resistance, and efficient fertilizer use, some future tools I can see that would help improve breeding and focus on these three main goals that Norman Borlaug had would be gene editing, as we're able to edit traits and utilize that in breeding. Also digital twins, so we're able to simulate the breeding pipeline and make decisions through simulations rather than through the field. Utilizing AI and machine learning in decision making, some of which I showed earlier, and also advanced phenotyping, because the better we can do our phenotyping, the better we can do our modeling. And just as my last slide here, I was asked to share some career insights. So these are just a few insights so far. Like Tabaray mentioned, I definitely like to take on new projects, and I've learned a lot from just taking on new projects and trying to learn just from different teams and different opportunities that are available. Taking time to innovate. So this is a picture that I took from a couple weeks ago, just walking through the field right before harvest. So I was taking notes on hybrids right before they were about to combine it. And I always seem to get new ideas while I'm out in the environment, walking corn, and really being integrated in the environments that I'm trying to make selections for farmers to grow in those environments. In this picture here, I have two pictures. On the top, right, I have an image of these were pioneer staff at the time in Algona, Iowa, and they are hand planting yield test locations. That was what they did at that time, is they hand planted. And now right below that is a picture of our modern day planter, of what we're currently using now. You can see there's no people involved in this. It's almost completely automated, except for someone driving the tractor. But in this image here, I'm sure these men planting this field could never imagine having a fully automated planting operation. And I can't begin to imagine what it would look like 50 years from now, of what planting or really anything would look like in breeding for 50 years from now. So it's always good to try new things, because you never know what is available until you do try it. And finally, networking. I'm a member of NAPB. I'm on the Borlaug Scholar Committee and really enjoy that. I always encourage graduate students to network both within the university and also other universities. And internships, I've learned. My first internship was with Pioneer, and I learned a lot through that internship, and really learned that was a career that I wanted to pursue. And now I'm working with some of the same colleagues that I interned for. So been Very thankful to give this presentation today, and I'm very thankful to receive this award. So thank you, and I guess if there's any questions, I can take those.
Okay, we have a few minutes for questions. If there's anyone in the room that has a question for Amy. Okay, I have to review my.
Thanks, Amy. I'm just curious, given your past experiences in the transition from analyst to breeder, how you see your role as a one breeder, two breeder these days, and how that's all this automating and designing the curriculum, but it's, yeah, I know you mentioned going to fields and taking notes, but anything about that, the role yourself as a field breeder in this time.
So I'm sorry, I wasn't really able to hear much. Just how do I see a role evolving from corn breeding?
Yeah, I was just wondering, given your past experiences, how you see your role as a corn breeder, as a field breeder specifically. I know you mentioned going to fields and taking notes, but anything beyond that in terms of how you see yourself in this role, this world of predictive analytics and automated feedback and so on.
Yeah. So in my first two roles within Pioneer and then Corteva, I did a lot of quantitative genetics work, and then also moving into some of the modeling work. But I still was integrated within the field. I was very lucky to be located at a research station where I was connected with breeders and also connected with the field still. But now that I'm fully just doing corn breeding, I really still enjoy working with some of that modeling work too, and incorporating that fully into my breeding program, and trying to be innovative in that.
Any other questions? Not, we're right on time. So, thank you, Amy. I'll pass it to Kat.
Yes, thank you, Amy, for such an inspirational talk. Unfortunately, she obviously wasn't able to attend in person today, but at a to-be-determined later date, she will be coming on campus to meet faculty and graduate students, so keep an eye out for that announcement. But in the meantime, Taba, can you please come accept the certificate and gift on Amy's behalf? Taba. With that, now I'll introduce our keynote speaker today, Dr. Arshnee Moodley. Dr. Moodley is a microbiologist with a PhD in antimicrobial resistance and zoonosis from the University of Copenhagen in Denmark. Based in Kenya, Dr. Moodley is a team leader for the CGIAR's Antimicrobial Resistance Hub at the International Livestock Research Institute. In this role, she works to reduce the risks and impact of antimicrobial resistance in agriculture, particularly in low and middle income countries. Please join me in welcoming Dr. Moodley.
Okay, I will try. And then feel free if I'm not doing it properly, because my husband's half Italian, so I tend to talk with my hands a lot. And so I'm not used to holding something in my hands. So I'll try my best. So firstly, thank you so much for inviting me to be here today. As I said, until a couple of weeks ago, I didn't know what this was going to be. I thought it was just me having an opportunity to share what I've been doing for the last 20 years of my life. And really the title of my talk today is this: it's feeding the world of fueling resistance. And I think this has been a topic that has been in my mind as I have progressed in my career. and also based on my workings with farmers in low and middle income countries and trying to balance their needs with my needs and my needs as a scientist. So the first thing is, I'm sorry to say, I'm not a plant scientist. I know nothing about breeding. I'm not a geneticist, but I am a microbiologist by training. So I understand a little bit of the words and the terms that have been shared today. But yeah, I'm really sorry. There's not a lot of plants in my talk. But nonetheless, I think hearing about Norman Borlaug's legacy and what he worked for, and I think what I do is a part of that. It's about improving food security, improving livelihoods in low and middle income countries, but in my case, through livestock. And this has been my journey for the last 20 years. As a microbiologist, I for many years had my nose in a petri dish looking at bacteria, watching how they grow and how they become resistant. And this is bacteria causing infections in humans or in animals, and how then do we best treat them? But and that's a picture for my PhD thesis from the University of Copenhagen, looking at that intersection between humans and animals and how we interact, not just companion animals, but also food producing animals. But then realizing that the world is bigger than my petri dish, decided to take my nose out of, and in some ways forced to take my nose out of the petri dish and enter the real world. And that's where I struggle. I still struggle a lot between my technical side, but also who I work for, who's my target audience, and how do I my research meet their needs, and how do we get to overlap and help them. And I guess the career highlight for me, and that's the picture at the bottom right-hand corner, was at the United Nations General Assembly last year, in September last year, where member states renewed their political commitment to addressing antimicrobial resistance, and that was taken there. So super happy about that. So thinking about Norman Borlaug and the Green Revolution and where we come from and where that started and where we are today. And I think in very much it's thinking about where we find ourselves when we think about our food systems and the different crises that we face. In particular, I work within One Health, and my entry point into One Health is animal health and how animals are viewed when we talk about human health as well as in environmental health, and particularly in terms of the food systems. And this is on the right-hand side. I think, can we do the pointer here? So this is an article taken out from the new Lancet Commission's report on food systems and looking at how food production and its impact on the planetary boundaries. I think it's a really fascinating report that has come out and really worthwhile reading if you haven't already done so. And so I asked the question today to you: if the first revolution was to feed the world, what should the next revolution be? It should be, how do we keep our food systems safe, fair, and within its planetary limits? So as we proceed today, think about that. If Borlaug's generation fought hunger, what is our generation, mine, or even the next generation? What is going to be your food revolution? And are we prepared to balance productivity with responsibility? And I think when we were thinking in the 40s about, producing food, I think it started the area of intensification. And I think we are now seeing the impacts of that intensification, and how then do we continue to produce food in a way that is both ethical, safe, sustainable, and all of those different words come into play. And that led me to think a lot about, based on my research and the work that I'm doing, is how are we building our food systems and how are they nourishing us, but at the same time, the paradox that they endanger us. And if by 2050, as our populations grow, we must produce more food, whatever they may be, animal products or crop products, but nonetheless, we need to increase our production. But that intensified, unsustainable production, and in my case, livestock production, drives the use of antimicrobials, which then leads to resistance, as well as environmental stresses. Both those practices impact the environment negatively, but as climate change progresses, that then comes back to impact the system. And so we have this very vicious loop that we find ourselves in. And how do we meet the rising demand for food, and in my case, again, animal source proteins, without accelerating antimicrobial resistance? Because animals get sick, plants get sick, you have to treat them, but that treatment leads to resistance. So how do we do that without selecting for resistance, which then has an impact not only on the crops or the animals, but on us as humans? And how do we do so within the planetary boundaries? So at the bottom, I have a figure from the Stockholm Resilience Center looking at the 9 planetary boundaries and where we are. So in 2009, we had only transgressed 2 of them. And now by 2025, we've had transgressed 7 out of the 9 boundaries. More so, and I think in the world that we find ourselves in today, is how do we produce food that is both equitable, built on trust, and still has resilience in that? And I find ourselves in a paradox, and I hope you are also thinking about those things as we move forward. So I work in a sector that very often is very negatively viewed in the world. And even in my own house, between my husband and I, he would like to get rid of all the pigs in Denmark, whereas I'm advocating for, well, we need them, and we cannot all become vegetarians or vegans. But there is a drive globally about a discussion on reducing our meat consumption. And so these are just some of the pictures that I've taken from newspaper clippings about, reducing our consumption. I would like to say that that's coming from a good place, because when we look at livestock production, it's very much linked to many different negative aspects, particularly when we think about the climate, but also when we think about pandemic and pandemic preparedness. And as I am based in Denmark and have a very strong connection to Denmark, as of last year, introduced the world's first FART tax. So an emissions tax that will be placed on our livestock producers as a way of meeting the Danish goals that have been set. But very often, I find my role in a lot of the global discussions is that it's very nuanced. Yes, in some parts of the world, we talk about excessive consumption of animal products. But in many other parts of the world, what we're pushing for is more production and more consumption. And these are the reasons why. And I think it's very important in that discussion, we remember the role that livestock plays in these different parts of the world. So livestock is linked very closely to livelihoods and an economy, and particularly around 1.3 billion people in low and middle income countries depend on livestock for their livelihoods, be it a few liters of milk to be able to sell so that you have money to send your child to school, or you have money to buy medication. These are the things, it's everyday life. It's not the 30 liters of milk, it's the 9 liters of milk or the few eggs that are produced. In many countries, it contributes to agricultural GDP. And as I said, livestock is not just economy, it's culture. It's linked to, it's a form of insurance for many communities in low and middle income countries, which you sell in times of stress, and when you need liquid asset, it's a liquid asset, and when you need money, you sell that, or you go into the production itself. The other one is linked to equity and gender. 2/3 of low-income farmers are women. And women control, and women who have this power, this economic power, despite the fact that culturally or in that particular country may not necessarily be the norm, but the money that they have, they put that into their household. They take care of the children. They provide food, education for their families. The other one, as I said earlier, it's linked to nutritional and food security. A lot of our proteins about 34% of our protein comes from livestock. It's rich in a number of nutrients. But we are facing many parts of the world food insecurity, starvation, malnutrition, stunting in children under 5. And so an egg or just a glass of milk really helps those children develop. And there is a growing demand. As economies improve in low and middle income countries, so do we see a change in diet and often a switch to animal source foods. But my work has always been on antimicrobial resistance, and more as we discuss the role of antimicrobials in allowing that intensification of our food systems. And one of the things I've come to realize in the discussions that we've had over the years is that we take antimicrobials for granted. we underestimate how much those antimicrobials have helped us to sustain or to increase our production, but also in terms of our own livelihoods. But then the converse is that if we use them, we create resistance, and resistance is bad. Resistance not only kills humans, it kills our animals, it reduces productivity. And so how do we balance the good of treatment? Because unfortunately, I don't think we can have a zero treatment policy. We would like to, but that's not possible at all. So we have to treat, but how do we treat in a way that we limit the resistance development and the spread of that resistance? So I like to say that antimicrobials is the invisible backbone, not just for animals. I've also come to learn you use antimicrobials in plant health. It's just called something different, and I'm learning how to translate or navigate the different words that I use in the different systems, but it is that backbone that allows us to have the systems that we have today. In many low and middle income countries, it's seen as a quick fix as a result of broken services, both on the healthcare setting, but also in veterinary health. And so these drugs, which are super cheap and work brilliantly, are used to uphold these broken health systems that we have. And on the far right, we have the political declaration from 2024. And in it, it says, member states commit to significantly reducing the use of antimicrobials in agri-food systems by 2030. I will say that we were hoping for a number, a target. A target is always good to have, but in the negotiations, certain countries were not happy about that number, and that came out. And so we ended up with something, a meaningful reduction, whatever that may be. So I ask yourself, in many parts of the world, in our animal production systems, antibiotics are given on a daily basis to our animals. And I ask you, would you take an antibiotic every day if your livelihoods depended on it? The other thing is that how do we ask farmers, based on the political declaration, by 2030, we have to have a meaningful reduction. How do we ask our farmers to reduce their use, but knowing that it's coming at a risk? And knowing that it's going to be linked to productivity and livelihoods. And so this is where my research has come in, or my work in the last years have evolved, is at this paradox that we find ourselves in, and how do we balance that? So before we can ask farmers to reduce their use, we have to ask, why are they using it? And so here, this is just a little causal diagram of our farmers, where we look at the different factors that are driving ultimately why a farmer buys a drug and puts it into an animal. And really, it's around breakdowns in the system. When you're starting from very weak veterinary services, not having a vet, the number of vets that are produced to the number of animals we find in low and middle income countries they far outnumber the number of animals there. And there's a really nice paper looking at density of veterinarians compared to animal populations. So already we have a stressed or burdened animal health service system. But at the same time, we have weak regulations. In many countries, the regulations may be there, but they're not implemented, or they don't even have the regulations at all. So you need both grassroots movement, but you also need very top level governance, strong governance, and the two have to meet. Economic incentives. I can produce, if I use an antibiotic, my animal is going to be robust. That antibiotic costs a couple of dollars. And I'll show you in a little while the cost of using an antibiotic versus vaccinations. And so at the end, if the motivator is a dollar motivator, those economic incentives, which we cannot ignore, when I ask a farmer to reduce their use, they're going to ask me why. What is the benefit to them? And I have to have an answer ready. And very often, the reduction of use of antimicrobials for farmers is not always for the farmer's benefit, it's for the greater societal good. And how then do we get them to do that, where the benefit is not directly them, but for their society? And that, I think, is a challenge and where strong governance comes in. Then we have other issues in terms of market pressures, biosecurity gaps, that all have to be addressed. But very often in low resource settings, we're not able to just build a better farm with better biosecurity. This is unfortunately not there. So how then do we work in the systems that we have to get the use that I want? The other thing is, how do we measure that antibiotic use? And I will say this is a statement that I put up, but I put it up in a way to aggravate myself so that I can tell you that that's the wrong statement to use. And even in the Lancet Eat Commission, this statement is there, that 2/3 of the global consumption of antibiotics is in livestock production. And this is what is driving a lot of the discussions around a reduction of that. But I will go one better and say, well, that's true. It is 2/3. But how big is an animal or that animal biomass that we are treating? And very often we forget the denominator in this. Antimicrobial use in humans nowhere compares if you want to treat an animal, a cow, which could be up to 1,000 kilos. So the amount, the milligrams of kilograms is very different. And so when we take into consideration the denominator, then things become a bit more interesting. And in some countries where we have data on biomass of animals, the human population treatment, very often we see that we use more antibiotics in humans than we do in animals. So this one-to-one comparison of volume is often very misleading. Although I will say to quantitatively measure antibiotic use or antimicrobial use in animals is a nightmare if you work in this field, as there are many different metrics to do so. And each metric has its pros and cons, and it's also dependent on why you want that data. So if you want to compare globally, then we would use the milligrams per PCU, which is the bottom level. However, it doesn't tell you anything about what is actually happening on a farm, in the field, because that's where you have to target if you want to make any intervention. But some of the other measurements require more data, and that data is often very, very difficult to get. Especially in a country like in Kenya, for example, where we're still working on a paper-based system. So digital is getting there, but not fast enough. And there are good reasons why they don't trust digital as well. So paper-based, you can't really do a lot with that. So this is the global database, or rather the global surveillance. It is conducted by the World Organization for Animal Health, and the program is called ANIMUS. So on a yearly basis, members, WOA members are not necessarily obligated to submit their antimicrobial use data. It's a voluntary at the moment. And so far, only 200 out of the 219 countries worldwide that do submit data to ANIMUS, only 55 make it publicly available. And I would say United States is not one of those countries that makes its data public. So far, the type of data that we get is largely import data. So if you're a country that's not producing an antibiotic, it's okay. You import everything, and that's what you produce. That's what you put into the data, into ANIMUS. However, it doesn't tell you anything about how the drug is used and why it's used. And sometimes it's also exactly what type of drug is there. But I will say that in the last years, ANIMUS has now been running for more than 10 years. They have seen a gradual reduction of use in different countries. And I think globally it's 5%, but in different regions, it can be up to 20–30%. So this is so one of the things I asked a poor PhD student of mine to do, Naomi, is that I want longitudinal data. So this girl had to unfortunately go to the field every 2 weeks, and you can see we started off with 73 farms. This is poultry farms. Every 2 weeks collecting data from poultry farmers, what they were using, both in terms of writing in a treatment book, but also in terms of depositing their use packages into a bin. So for 8 months, every 2 weeks in the field visiting. So she didn't like me very much. I will say that we have gotten the best data, and it's a very unique data set, both because we have multiple consecutive production cycles, and we have very granular data on what is used, when it's used, and also how it's used. I would say it was a very expensive study as well. It was about $250,000 to do. But nonetheless, I think it was money well spent. So basically what I want you to take away from this, what we're seeing here is different drugs. So each column is a different antibiotic. And then the little bars within each column is the different periods within the production cycle. And I was quite shocked to see how much antibiotics are given and how frequently they're given within a production cycle. And that's where, in some cases, this is broiler birds. So they're 6 weeks old. Some birds are treated every day. Every day you get a little bit of antibiotics. Whether they need it or not is a completely different question. And then we're not able to dissect, is it because, does treatment the birds mean they don't get sick, or are they being treated because they are getting sick? So we don't know which comes first. However, what we're able to know is, or to be able to look at based on that granular data, is how are they using it. And so ideally what you want to be is in the green bar, which is both the dose that you're given as well as the frequency of that dose being given. And you can see for the most part they do it correctly. But there are some cases where they're overdosing, or they're giving a very high dose for a longer duration or a high dose for a shorter duration. And there is some underdosing happening. And all of this is leading to resistance development in that population. Thankfully, it's broilers, 6 weeks in and out. But imagine if you're a cow or a layer and you're there for a year, 2 years, or if you're a cow with mastitis, and typically in these systems, they keep the cows for 6, 10 years, and you've been treated. How much of antimicrobials are in there? Noting that they don't respect withdrawal periods. So how much of that is entering, of residues is entering into the food chain? Nonetheless, I think we're scratching the surface on this data and how to put that out. But coming back, so the political declaration says we have to reduce our use. But the question is, how low is ever going to be enough? Some countries set targets. So in Denmark, the target was 20% by a certain, the minister said 20% by X date. And so the entire country and the entire production system geared towards doing that. And in Denmark, you can. It's 6 million people, lots of good resources, lots of good databases, lots of good incentive, high trust in government. And so we were able to have that reduction. But the question is, what reduction will be enough for a meaningful impact? In some countries, we have had a 20% reduction, but what is it that we're measuring and what is the outcome of that? And very often that reduction is very much going to be linked to the circumstances in which that has to happen. And whether or not any reduction will, if it has a negative impact on production and livelihoods and food security, should we still advocate for that? And so I think we haven't quite gotten to a number yet. A number will come at some point, but I think it's very important that that number is, we will have a global number, a global target, but very often, how do we translate that to different circumstances and instances, and whether it has to be linked to certain species or not. And I think that this is a discussion that we're still not having, but hopefully we'll do. So one of the things that we look at in that data was what are farmers mainly using. So growth promotion here in the US has been banned. Also in Europe, it's been banned. And in many other countries, they are trying to ban it or phase it out. The legislation may be there, but it's difficult to implement. So one of the products that we see is something called Alicerol. And Alicerol is a multivitamin that contains 4 antibiotics, including colistin. Colistin is considered a last resort antibiotic for the treatment of severe multi-drug infections in humans, gram-negative infections in humans. Yet it is included in this product here. I will say it is a product that is produced in the Netherlands, which is part of the EU, which do not allow these products to be used. And so I have very many moral, ethical questions on this. And then when you look at the productions, the manufacturer's website, this is what it says. It's a product that stimulates egg growth, increases growth, improves feed condition. And if you're a farmer, who doesn't want that product? So it's one of the most used products that we see. So very often in the discussions, when we talk about reduction of use, we talk about increase of vaccination. And that is part of the new mandate also from WOA, and that is also put into the political declaration. All countries must have a vaccination plan for animal health by 2030. So just this very simple question, I asked one of my students to please go to the agrovet where you buy your products to compare how much would it cost to buy Alicerol and treat your flock versus vaccination. So Alicerol, 100 grams of Alicerol is $5. And with that $5, we can treat either, if it's prophylactically, 1,000 birds. If it's for therapy, it's 500 birds. Now, broiler birds tend to get infected by Newcastle disease, is one of the 3 main poultry diseases. If you have to use the subsidized vaccine, so this is in Kenya. it will cost for the treatment or for the vaccination of 1,000 birds, $35. And very often when you do vaccination, because you stress the animal, you also have to give multivitamins and sometimes a little bit of antibiotics as well. So that increases the cost. If you use a combination vaccine, which is for 2 diseases, plus it's now an injectable vaccine, you have to have a vet come in to do that work for you. So 1,000 birds will now cost $140. So if you were a farmer and you're worried about profits, so profits are one of the important things of your business model, are you going to go with, is it cheaper to vaccinate, or what would you choose? Antibiotics, $5, or sometimes over $100 to vaccinate. But you still have to treat anyway. It's not a guarantee that you can get other diseases. So one of the things is that, antibiotics are really cheap and they work really, really well. And so what is the incentive for farmers to not using that? And also when you look at the production costs, antibiotics are a very, very small percentage. It's less than 2%. The biggest cost is feed and then the inputs to that system if they have to buy new animals. So how do we make antibiotics a less attractive option for farmers with not many resources? And that's where I am right now. And how do I break that? So this has got me thinking about these loops and how does we think about expansion of our livestock, because in low and middle income countries, we have to increase our production, but we're doing so in circumstances where in the absence of good governance, how do we do so it will be an unsustainable intensification. And so this is then the conundrum linking between misuse of antibiotics together with resistance development, but there is also land use changes. That expansion of our livestock production is leading to deforestation, which is leading to biodiversity loss, which is impacting the amount of water availability. But this is also linking to climate change, and climate change is either the driver or a sort of a negative onset of that production. And how does all this come to play? And linked to this is this climate vulnerability, which is also driving a lot of the disease episodes and the commingling that we're seeing. And so how do we then think about that in sustainable livestock production when all of these different crises are happening at the same time and different policies are being implemented or put into place to try to protect them in isolation? But I think, how does it all come together in that complexity? So this is the only slide I put in. I snuck in just a couple of hours ago about plant health, because I think very often it is we say animal health is neglected because it comes human health, then animal health, and I guess plant health is somewhere at the back there, and very much a neglected area within the One Health space. But the plant people are very good at making sure that their voice is heard. And so every once in a while, we'll have one plant person in the room that will say, don't forget about the plants. And what I think is truly interesting is that antimicrobial resistant genes, the way we see that in a clinical perspective, has an origin in soil bacteria. And typically they're mobilized from the bacteria or the organisms that produce that antibiotic. They've just been mobilized into pathogens, and that's why we care about them. But very often the antibiotics that we put into the environment, either through our own practices, be it animal production, or our human use of antimicrobials that then end up in the environment through our waste, as well as the antibiotics that I've come to learn that you use also in plant health, you just call it something else, together with heavy metal-based compounds, they co-select, or you have either cross-resistance or you co-select for resistance in microorganisms. And those microorganisms persist. I think very often we don't really know what would be the equivalent marker. We tend to use E. coli because E. coli is a really great organism, but I've often asked my plant and environmental enthusiasts whether that is a really good indicator of environmental health. And very often we see that vegetables do contain residues, both in terms of the antimicrobial residues, but the antimicrobial pathogens themselves, which then either stay in the environment or we as humans acquire them as we eat these different foods. And so when I think about now, when we talk about stewardship and that rational antimicrobial use, is how do we bring in the plant angle to that? And I think that that's part of the work that we have to do moving forward. And that leads me into the next part of our work, my work, which I think resonates a bit more with what I think you're doing here, is this interplay between antimicrobial resistance and climate change. So this was an article that came out in Lancet Public Health earlier this year, trying to look at, well, what evidence is there? Does antimicrobial resistance drive climate change, or is climate change driving antimicrobial resistance? And here we can think about temperature changes, pathogen distribution, selection, but at the same time, climate change and the processes related to climate change are microbial processes. So if we have antimicrobials in the environment that are changing the microbial composition, then there must be antimicrobial resistance, or antimicrobials themselves in the environment are also driving climate change. And so it's a really nice paper looking at terrestrial systems, aquatic systems, and looking at how one drives the other, but at the same time, what are potential trade-offs, synergies between adaptation or mitigation strategies. So again, chicken or egg, what comes first? I would really recommend that. And this is where my foray into the environmental sector came in. And I think this is Chebet's work, and Chebet is a PhD student of mine, trying to bridge that gap between animal health and environmental health. And the question that I had is that we treat our animals with antimicrobials. Those antimicrobials end up in manure, and this manure then ends up on is then used as fertilizer. Particularly, we don't use a lot of synthetic. We don't actually use synthetic fertilizers. We want to use the manure there. And so what she was doing was setting up experiments to look at treated manure, so manure with different antibiotics at different concentrations, using antibiotics we use in livestock, and looked at greenhouse gas emissions from the manure heap, but also looking at the nutrient quality of that manure. And basically what she showed was that when you have antibiotics, irrespective of the type or the concentrations, it increases greenhouse gas emissions. But at the same time, it affects nitrogen cycling. And nitrogen cycling is an important component, because especially if that manure is going to be used on cropping fields to add nitrogen to the cropping fields, and basically showed that actually in some cases, it had a negative impact on that nitrogen cycle, and that led to either leaching into the environment and potentially reducing soil fertility, but at the same time, other nitrogen losses through air, for example. And this is really critical, as I said, when we think about the utilization of manure as fertilizer. And then Fred went one step further. Fred said, well, okay, how do we connect all of that? So Fred's job was to, part of his PhD was to take samples from the animals, samples from the manure, from the compost heap, and then from the fields that were amended with manure from the farms. And we're lucky because we have these integrated systems in Kenya. And so he was looking at levels of residues, antibiotic residues in these different samples, and then trying to estimate the ecotoxic effect of them. And so obviously it really depends on the antibiotic, and its decomposition will affect how much of that antibiotic you find. And particularly, we'll see the sulfurs and the trimethaprins, they tend to last longer. They don't degrade as fast as some of the other antibiotics. And so depending on the system that we have, you will find it, but the ecotoxic thresholds are very minimal depending on the antibiotic. And I think the key takeaway, and I think it's very much linked to how we use and the amounts that we use and the type of production systems, we don't have the intensive production systems like we see in Europe or in America. It's still smallholder. It's 1 to 5 cows, maybe 200 to 500 broiler birds. So the production system is still low. And so the use of antimicrobials, even though imprudent, is not as high. So what is coming out of the animal health production is still some residues with some ecotoxic effect, but not as bad, but definitely an area to think about. So when we think about this vicious feedback loop, and I think, again, coming through how we're thinking about our food systems and the practices in one area and the negative consequences of those practices, how is it all kind of driving itself? And this is what I see in real time as part of our work in these countries, is that yes, we're hearing more about disease outbreaks. And even in Europe, now we've just had an FND outbreak in Germany. We haven't had FND for a very long time. It's a trade issue when you have these types of transboundary disease. And partly it's relation to how vectors are changing. Vector distribution is changing. So we're seeing disease incursions in Europe that we haven't seen for 100 years, or we've eradicated, and truly now testing our surveillance and risk mitigation measures. But that's leading to different strategies, and in some cases more antibiotic use, not because they're bacterial pathogens, but because they might be viral or some other parasite. But what farmers have access to is antibiotics, so they end up treating it with an antibiotic in any case. This leads to more manure residues in manure, which is going into the environment, which is disrupting both methane emissions, but as well as nitrogen cycling, which is then leading back to a selection of pathogens, which is then leading back to more outbreaks. So I think this vicious loop is what we should be concerned about, and how, when we think about the future food systems, is how to break that, or how do we best do it in a way in which we don't have these kind of incursions coming in. And this is also related to the pandemic agreement, which took many years to negotiate and was finally approved by member states in May, which again is coming through, and where animal production is a big part of that when we think about preparedness for the future. So I guess the question is, are we treating symptoms of a broken food system, or are we addressing their root causes? And if climate stress fuels antimicrobial resistance, and antimicrobial resistance worsens climate change, where should we intervene to break this loop? So this led me into another area, and I come back to the question I had earlier about how do we get farmers to change their behavior? And at the end, it really is about behavioral change. And so when we thought about, well, what is it that we want farmers to do? And what do we want them to do better? And the first question is, what is desired behavior? And so we took an FAO document and we dissected that. And I worked with a human behavioral lab. They're based in Cambodia, but nonetheless, they came to work with me in Uganda. And we took that FAO document and broke it down. And essentially what they came back with is that if you wanted to use antimicrobials in a prudent way, ultimately that meant changing 34 behaviors. That's 34 behaviors that have to have something about knowledge and the capacity or resources to be able to have that behavior change. And that's, I think, is a tall ask of anybody to change 34 behaviors. And so as part of that work and a part of this foray in One Health and the interdisciplinary nature of or the ability to work with different disciplines, I think is where the gravy or the sauce comes in, and that good part. And I must say, as a microbiologist, know nothing about social sciences. So it was probably 2 years of the worst time of my life trying to understand what social sciences talk about. I now feel like I understand it a bit better. And to be honest, I still like ones and zeros, black and white. My husband is a historian and a philosopher, and he says the world is very gray, Arshnee, and you have to get on board with that. So in that behavioral analysis of the 34 behaviors, we went to ask our farmers about whether or not they're either conducting that behavior, and if they're not conducting it, why? And do they have the knowledge or the risk that is associated with the knowledge that by not doing that, there's a risk with that. And then whether they had the means to change and adopt that behavior. And what you'll see there is a lot of reds. And a lot of reds means they're not doing it. A lot of yellows means they're kind of doing it, but not very not a lot of times, and very, very few greens. And one of the major barriers for behavior adoption is affordability. And a lot of it requires effort. It's usually structural efforts that have to be built, and a farmer doesn't necessarily have the resources to do so. If you're a female farmer in these settings, you also don't have the financial capital because you don't have access to that finance to do that. The effort is always more. And knowing myself, if anything requires me to do more, something that is just demanding, it's very hard to sustain that behavior. And so the incentives to change has to be very, very rewarding. Then comes the lack of knowledge, is that what we're asking farmers to do, they might not necessarily have the knowledge to do so. So how can I demand them to stop using antimicrobials when they don't necessarily have the other resources about feed. What is good quality feed? How much feed I should be giving? How should that feed be stored? So those are all of the things that I think we need to take into consideration when we're asking an individual to change their behavior. And very lastly, gender. And again, coming from a black and white world, ones and zeros, I never really thought about gender. In living in Denmark for so many years was also a very flat structure. But coming from South Africa, always rebelled against, I don't know how to cook. Which is one of the things I think my mother is very upset about me, I never learned how to cook. I despise cooking. I love eating food. So I had to marry somebody who's also was a good cook, and he's half Italian. So that worked out really well. So I asked ChatGPT, and I actually asked it again last night, because now it's 5.0 is the model, and this was at 4. Draw me a picture of a livestock farmer. And it drew me this gentleman on the left. And then I said, no, I want a livestock farmer in Africa. And it drew me the other gentleman on the right. And so we're very biased when we think about a farmer. And if you close your eyes and you think about a farmer, I would bet not many of you think about a female farmer. I could be wrong, but I think stereotypically they're males. But as I said very earlier, 2/3 of our mixed crop livestock farmers in low and middle income countries are women. And when I looked at data from Europe, it's only 30% of the farms that are managed in Europe are female. And very often when we think about our intervention, we think about our intervention in a very technological way. And we don't think about, well, how are they going to access that? How does a woman take care of that? Women typically don't keep cattle. Women typically keep poultry in low and middle income countries. And so if we have intensification, when we think about intensification, we often tend to exclude women because women have other chores in the household. And so we push them out of business. More than that, they not necessarily have access to vaccination. It could be that if it's a male veterinarian that has to come in, he's not allowed to enter the farm if it's a female there. And those kind of social norms have to be taken into consideration. So as part of this adventure, and again, another adventure into an area that I knew nothing about, was working with the Global Strategy Lab in Canada to come up with a gender research framework for livestock. So if you're interested in gender, livestock, and AMR research, please go and have a look at our paper. And it's really asking, how do we think about our interventions in a way that is gender sensitive? So again, now a question: whose knowledge and labor sustains our food systems, but whose voice shapes the rules for that system? And can we have stewardship without social justice? And I think the Lancet Commission's paper is also talking about social justice when we think about the food systems. So very biased, of course, is the Danish lesson about antimicrobial reduction in the livestock sector. And I say we because after 20 years, I feel that part of me is Danish now, and I get the jokes, and I love smørbrød. And I can say some of the words, pronounce them correctly. So 50%, we've had a 50% reduction in our antimicrobial use in the livestock sector, and particularly its pig production. We produce 35 million pigs a year. We're only 6 million people. So it's a big sector, and it's a big part of the economy. We've banned growth promotion. We have various incentives that retract or make antibiotic use less desirable. We have robust anything. We are all bound by a social security number. It's called the CPR number. You don't exist in Denmark if you don't have that. So you cannot thrive. You cannot live in Denmark without a social security. And everything is tied to this number. And it ties for the rest of your life. You can look at your entire records from the day you were born. Every treatment you've had, doctor's visit, lab result is on a database, and you can access that. So we're very well surveilled, and our information is there. So that allows us to do some really cool things because we have all the data and all the systems are linked to one another. So we've consistently had the lowest amount of antimicrobial use in our food production sector. We also have the lowest amount of resistance in our food sector. But despite all of that, and despite all of the major changes that we've had, what we haven't been able to show is that by reducing our use means we have less people dying from a resistant infection in our hospitals. Because that's ultimately why we are asking for a reduction, is because we want less people dying from resistance that they acquire through the food chain. So we haven't been able to do that. And that's because we live in a soup in a soup that is not clean or anything like that. We've already shared our resistance. We're all sitting here with resistant bugs floating around, and we're exposed to it both by our travels, the food we eat, the company we keep, the animals we love and take care of. We acquire that, and we give just as much as we take. So attribution is very, very difficult. And also the impact is often very delayed, and typically research projects never last that long. But thankfully, the Danish state very much is involved in resistance and continue to fund the research, the surveillance programs. So again, in a country like Denmark, despite all of the initiatives that we've had, is how do we show that that reduction is leading to a reduction in human infections? And so pretty much, again, how do we do that in low and middle income countries? So I'm almost finishing up now, and I'm trying to circle back is how do we feed the growing world amidst these vast inequalities or inequities that we have? Production systems are very different. Just in one country, it can be, yes, we have intensive, but we have pastoral systems as well, and we have extensive systems, and resources are very different. And so this global discussions have to be somehow distilled and made relevant to a national context, but it still needs to be married to the global context. And so this is what a balanced production system looks like for livestock. And it really is all of those different elements, from stewardship to welfare. Animal welfare is something we don't talk very much about and should talk more if we make animal welfare as a core reason of productivity, linked to resilience, linked to climate, linked to gender. But very often that production has to happen within a much bigger system, and that requires different pillars. That requires good governance. So we need laws, we need regulations, but that has to be linked to incentives, and not just market incentives, but other things or incentives that disincentivize usage of certain things or different practices. Global coordination and always surveillance and data are really important, but all of this is pinned on trust and transparency and equity. But at the end, it all comes down to individual behaviors, unfortunately, and how then do we have that behavioral change? Yes, we need innovations, we need new technologies, but that's not enough. We can produce it, but if a farmer doesn't want to adopt this fancy vaccine or probiotic or whatever they may be, who cares? Who cares? And I think very often I find that as an academic base at the university, I often don't have, and I like what Amy says, is going into the field, getting to know who you work for, and there's those targets that allows you to be able to think about the work that you do and to frame it in a better way. So we need all of this, and because technology is not going to lead to adoption, and how then do we balance that in the context in which we're doing so? So I'd like to say that, the world that we're feeding now is not just about that intensive production and more calories on a plate or just calories on a plate, but I think to do so in a discussion that is, fair, safe, and sustainable. And how do we do that? Do we maintain the status quo, or do we need another type of revolution? So I'd like to also, coming back to what Amy said, 50 years from now, what will people say we did? Because it's our time. And did we act with the evidence that we have? And what will we be remembered for? Did we feed the world, or did we break the world? So I want to say that, at the end, I get to come and say a lot of great things and experience a lot of wonderful things, but I'm coming on the backs of all of my wonderful students who have produced all of the data and wrote many of the papers that brought me here. So I just wanted to give a shout out to them. And to thank them for fueling my career, and it's also been such a privilege to work with them. So I just want to end with that. Thank you very much again. Bye.
All right, do we have any questions?
Hi, my name is Lovepreet Singh, and a nice talk. And I was thinking about your comments about the production, the goal, which what we're using the antibiotic use from United Nations. But it seems like it still can't get a figure or is still meaningful reduction which can be maintained. And also I was thinking that are you thinking about low versus high income countries because the priorities are completely different there, and in terms of as you mentioned, resources are completely different. So in terms of those goals, are they different for lower versus high income countries? Do you think like in this space, maybe high income countries can be more like leading the situation and be more contributing towards reducing the antibiotic consumption versus lower income countries because they have those struggles there that they're so any thoughts on that?
So when we look at, so yes, we need global goals. I think it's important to coalesce around a global goal to get everybody on board with that. But we need national targets. In any case, how we translate a global goal to a national target, we don't really know at all, depending on what country it is. And I don't know if in the United States you have a national target. I'm not sure about that, but we do for some countries that they do set that. And that's really important to have a baseline to work toward or some target to work towards. Now, when we talk about low and middle income countries, what they use is nothing compared to what we see in intensive productions in high income countries. Asia is different. I'm talking about Africa now. Asia is completely a different scenario. What we see in Africa is use is very low, but the number of people that die from an antibiotic reduction or from an antibiotic resistant infection is the highest in sub-Saharan Africa. It's 90% of the global deaths. And I think right now it's about 5 million that is both a direct result of a resistant infection or attributed to a resistant infection. 90% of that is largely in sub-Saharan Africa. But who should pay for that? We're not necessarily, the reason why a lot of people die because of access to an antibiotic, they may not have that. And this is a discussion on the global pharmaceutical industry and where they put their product. They want to say, we're going to put a product where it's not going to be used or it's too expensive and patients cannot afford that. So that's also another issue that we have to bring into perspective. I think the challenge is how do we get the strong global South voice in the global discussions? And that's been one of the difficulties in this global discussion. But so I don't know if you recently saw that I think it was Fiji that won a court case in the Hague that says that you can start suing other countries for problems you have in your own country, and this is in links to climate change. I don't know if you saw that ruling from a couple of months ago, and I was thinking whether that could be the same as well. We're thinking about who are the polluters, and in this case, who are the users of antimicrobials that are driving resistance that potentially have the propensity for global spread. Can you then sue another country because of that? I don't know.
In crop management, we also have a problem with resorting too much to antifungal applications when we have good breeding options for resistance. I don't know if it's been considered much in animal breeding circles. I know the organism is much more complex, the experimental units are a lot more expensive and so forth, but is there any attempt to use resistance breeding and genetic and more to reduce
So from what I'm not an expert in animal breeding, but this is part of it to breed animals that have some reduced or rather resistance to certain pathogens. I think very often it's coming at a disadvantage to something else. Not always the case. And now what we're seeing is new emergence of either old disease, old pathogens, or new ones. They don't fare very well. And I think in our context, which you have yourself worked in, from what I heard, the exotic breeds don't fare very well in our limited resource settings. And I think it's the indigenous breeds that do better, but they're not necessarily the high producing animals. And so the question is a balance between that. And I think very often it's the trade-offs and whether or not, and I think also saw a little bit of trade-offs in Amy's presentation, is in one context, one is better than the other, but in a different context, it may not fare so well. So there has been some work in that.
Any last questions? With that,
I just want to end, but when you go to the reception and you're thinking about the food that you're eating, maybe you want to think about where that food came from and what's in it. Thank you.
Yeah, like before, as a token of our appreciation, we'd like to present you with this certificate and gift from France.
Thank you very much.
Thank
Thank
you.
you. Thank you.
Some final thoughts just to wrap this up really quickly, because I think we're all tired and hungry and ready to go. Thank you so much to Dr. Jacobson and Dr. Moodley for being here today. Your insights and dedication are truly inspiring. Today's lecture was a wonderful tribute to Dr. Borlaug's enduring legacy, and we extend our heartfelt gratitude to everyone who made this possible today.